Bantala. So, hello everyone. Good evening. Welcome back, not to the online master course, but to the PYQ series of 2022 plan breeding. तो आज हम पढ़ने वाले हैं plan breeding. So we are going to discuss plan breeding questions today. Hi. Good evening. Good evening. Good evening, Nutan. Good evening, Preeti. Good evening, Dhruv. Good evening, Siddharth. Good evening, Vijay. Will you read it, sir? What shall I read it? क्या पढ़ना है मुझे? स्लाइड मिस हो गया था क्या? समझा नहीं ध्रुव क्या बोल रहे थे? हाय मधु, गुड इवनिंग. ये सिंचन कौन है? क्या नाम है तुम्हारा? You are walking today. Will you read it? You are walking today मतलब समझा नहीं क्या बोल रहे हो? तुमने पहले will you read it? अच्छा क्वेश्चन का बारे में पूछ रहे हो? So you are talking about the question. Yes, Ankit, good evening. Yes, फटाफट जल्दी ऑन कर लो. क्लासेस स्टार्ट करने वाले हैं. Like we are going to start the plan bidding discussion. तो बहुत ही crisp, solid 30 questions है जिसको आपको पता होना चाहिए. Like you have to be very much aware of this 30 questions. So we'll be discussing the 30 questions. And if you are able to see the pattern of PYQs, so the questions have been frequently repeating. Like whatever questions that we have discussed previously, more or less like similar kind of questions have been asked every year. So like if you just cover these PYQs at this very end moment, so at least you will be able to cover the subject 30 to 40%. I won't make the fake promises that you will be able to cover like 90%, 80%, but just by covering the PYQs, you will be at least able to cover 30 to 40% of the entire syllabus. And if along with the PYQs, you are going to attend the marathon, anywhere, somewhere between like 60 to 70% of the total syllabus will be covered. Sir, did you take the class today? Yes, I'll take the class today. I'll be teaching the plan bidding for all of you. मैं सिखाऊंगा या आप लोग मुझे सिखाओगे प्लान बिडिंग? कौन सा सही रहेगा? So like Abhishek was pretty much upset. थोड़ा upset था, like he was not willing to take the class for you people. पूछो क्यों? So like Sinchan, like I wanted to know your name. क्या नाम है तुम्हारा? सिंचान क्या है ये? We will teach you. अच्छा, तुम लोग पढ़ाओगे, सही है. Okay. So back to the point. Your plan bidding mentor Abhishek Sir was actually pretty disappointed with you people. I feel like men should stop teaching. I hope like you understood like why I said this. क्यों बोला ऐसे मैंने? So can you guess? मैंने बोला like men should stop teaching. क्यों? Viewers, अच्छा, point पे आ गए. Is it because of the views? Yes, of course it is because of the views. Students like even like I was not very much happy because of the views like that we were getting for the plan bidding PYQs. But why? Because first video like 2020 like PYQ videos like there were more than 2000 plus views that he got, but suddenly I don't know what happened from 2021 paper that but the views significantly dropped. So why I'm here not to just ask you that watch more, but I am here to tell you that these PYQs are very much important. I don't know like whether, see, it's totally fine you attend live or you take the recording. It is totally fine. It's up to you. But if you are missing it for one or the other reason, thinking that it might be the waste of time, or you might feel that you already know everything, or you are not getting the time, or you're postponing that to the upcoming days, then you are definitely doing a mistakes. See, this is a flow that we are keeping you, or we are making you to go through. Like if you're not going through the flow, later you will not be able to cover everything somewhere, or something or the around, like you'll miss. Okay, so I would definitely suggest ki in the last moment, like since you are left out with like last solid 30 days, like leave the last 3 days, next 3 days of next month, this solid 30 days, like you have to fine tune yourself, makes yourself so much productive, and involve in yourself in times of like rigorous revision. You have to like watch all these videos, whether you watch it in 2x, 1.5x, 1.25x, that doesn't really matter. But everything that matters is like you have to properly analyze PYQs, because at least you can expect like 20 to 25 questions more or less like directly asked from the PYQs only. So starting from 2020 to 2025, like if you are able to cover it, so minimum, tell you, like at least you will be able to cover 20 to 25 questions directly without spending too much of time on the questions. So you should not neglect any of the PYQs. So from 2020, since the pattern has changed, like from there to till now, like if you have seen some actual pattern, if you have seen the trend, common questions, the easiest ones are there, that they are repeated like every year, right? So you should not miss them. Sir, when will your class start? Agar this is what you, like this is how you people are watching it, and this is how you people are consuming it, I don't think like I'll be able to start my classes. I won't be able to start it. So where is the motivation? If not you people, then who will motivate us? और हार्डली लाइक जैसे कि मैंने देखा था लाइक I have seen the comments also, like hardly like 1 or 2 comments, or 3 or 4 comments have been posted in that previous video also. If you know like this PYQ questions have been already discussed. अगर आपने YouTube video open करके देखा है, like in the previous years we have already done all the PYQ sessions from 2020 to लेकर 2025 तक. But again we are spending our energy, हम again वही videos को दोबारा कर रहे हैं. क्यों? The only difference is with the smart board. पहले हमने सारा लैपटॉप में कराया था, now we are doing it on smart board. We thought you people need it, so that's why we are again spending our whole energy. But if even after doing that, if you're not satisfied, if you're not watching it, see, if you're not satisfied, let us know. If you have any feedback, let us know. But if everything is perfect but still you are not doing it from your end, that's not a good thing. Abhishek sir should not be upset. He still need a marathon. This is because of the views watching. Okay. So that don't say that you are the only reason, that's why we are here. Are, see, of course, like let me be very honest, like I am here only because of you people. It's same for me, just like it's for you. आप लोगों के वजह से ही मैं यहाँ पर हूँ, वरना यहाँ दाढ़ी बना के एयर्स लगा के नहीं आता, कि सब कुछ दिमाग खराब करके नहीं बैठता यहाँ पे, आपके सामने नहीं आता हर दिन. ओके, मैंने एक target banaya tha. Like I made a target, and let me tell you this also. I made a target. See, the day I complete the seed science and technology, then I'll come in shape again. So till then, like until I do not complete the seed tech, I thought of like punishing myself, so that I'll be reminding myself कि हाँ कुछ ना कुछ है जो मुझे बहुत जल्दी खत्म करना है, because all these things are completely irritating, बहुत irritate कर रहा है. बट स्टिल वो द ओनली रीजन मैंने सोच के रखा था, क्योंकि यहाँ पे बहुत सारे डिस्ट्रैक्शंस है, बहुत सारे अलग अलग काम पे मैं जाता रहता हूँ, लाइक बहुत सारे like so many things is going on in agri edit. But still, to keep myself motivated, to keep myself reminded, because you people are waiting for the book, तो मैंने इसलिए ये गेटअप लगाया है. like hopefully like in another 1 or 2 days like things will be sorted, so I'll be giving the good news for all of you. But still उससे अच्छा कि थोड़ा तब तक तो मुझे ये सब मानना पड़ेगा. Sir, we even ask Abhishek sir that we really wanted him to give us the marathon. Okay. Breeding handbook come. अरे भाई मैं सीटेक नहीं खत्म हो रहा है, एंड प्लांट पैथोलॉजी इज लाइक, प्लांट पैथोलॉजी के बारे में बात करूंगा तो मूड ही ऑफ हो जाएगा मेरा. अभी मैं बताता हूँ उसका, फिर कभी बताता हूँ. ओके, यू सीम राइट व्हेन यू लर्न. ठीक. ओके गाइस, लाइक दैट्स ओनली रिक्वेस्ट कि थोड़ा बूस्ट करो, खुद को भी बूस्ट करो और एग्रेड को भी बूस्ट करो, अपने मेंटोर अभिषेक सर को भी बूस्ट करो। मेरे को बूस्ट जरूरत नहीं पड़ेगा क्योंकि बहुत सालों से बूस्ट करते 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 आ रहे हो, वो भी मेरे पास स्टॉक में पड़ा हुआ है। अगर आप टेम्पररी अभी बूस्ट भी नहीं करोगे मुझे तो चल जाएगा, तो मैं तो करता रहूंगा, बट बाकी मेंटर्स को बूस्ट चाहिए होगा, जैसे तुम लोगों के लिए बूस्ट चाहिए। सो बूस्ट हिम, good, keep some comments, like appreciate him, and also like give your views, interact more, and most importantly watch all the PYQs. Okay, not only ऐसा नहीं कि पारुल वाला सारा क्लासेस देख लिया बाकी को छोड़ दिया। प्लांट पैथोलॉजी सिर्फ नहीं आया, सिर्फ प्लांट पैथोलॉजी नहीं आएगा एग्जाम में। एग्जाम में प्लांट ब्रीडिंग भी आएगा, जेनेटिक्स भी आएगा, बाकी माइनर सब्जेक्ट्स भी आएंगे। अगर आप एक सब्जेक्ट में प्रॉपर हो और बाकी सब्जेक्ट में आप छोड़ रहे हो और इग्नोर कर रहे हो, रैंक में बहुत इफेक्ट करेगा, राइट? तो देख लेना, ठीक है? थोड़ा पारुल मैम के अलावा भी बाकी क्लासेस को भी देख लेना, पारुल का भी देखना है। वो तो ऑलरेडी मैं बोलूंगा नहीं बोलूंगा फिर भी आप लोग देख लोगे, बट हाँ बाकी क्लासेस का भी देख लेना आप लोग, ठीक है? टेल मी व्हाट हैपेंड। अच्छा, ओके गाइस, सो दैट्स इट, सो लेट हिम स्टार्ट द क्लास। बहुत 30 क्वेश्चन ही है, थोड़ा सोचा मैंने तुम लोगों का थोड़ा ब्रशअप कर दूं, फ्रेशअप कर दूं तुम लोगों को, बहुत टाइम हो गया इंटरेक्ट करते हुए। So I'll be back very soon. I said don't be disappointed. So we are here for you people. Like we are doing all of these things just for you people. Some again I'll come after this breeding session. Next session will be genetics. Genetics का 2020 से लेकर 2025 तक सारे सेशंस कराएंगे। सीड सेंस का 2020 से लेकर 2025 तक सारा सेशंस होगा। उसके बाद ब्रीडिंग का मैराथन होगा, प्लांट पैथोलॉजी हो गया, ब्रीडिंग का मैराथन होगा, सीड सेंस का होगा, जेनेटिक्स का होगा। Okay, don't worry about these things. Like we will do it. If we see someone is asking like if we so what's up with your hair के बारे में बोला था ना कि एक टारगेट बना के रखा हूँ कि सीड सेंस जब तक कंप्लीट नहीं करूंगा तब तक सैलून नहीं जाऊंगा, क्योंकि ये सब इरिटेटिंग है। तो इरिटेट जितना करेगा उतना जल्दी मैं पुश कर पा रहा हूँ खुद के लिए, क्योंकि यहाँ पे अलग 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 काम होता है ना, तो मैं ज़रा थोड़ा डिस्ट्रैक्ट हो जाता हूँ। तो ये चीज़ मुझे याद कराता है कि हाँ बुक है, मुझे खत्म करना है, इसलिए आई विल नॉट मिस इवन वन क्लास ऑफ प्लांट साइंस विद ट्रस्ट अग्रेड अ लॉट। ठीक है, अग्रेड को ट्रस्ट कर रहे हो तो खुद को भी ट्रस्ट करो और ट्रस्ट द प्रोसेस, and thank you so much for trusting, it matters a lot. And if I watch all the classes, is that enough for the plant breeding? देखो इस लास्ट मोमेंट में मैं तो सही, ऑफ कोर्स लाइक इन दिस लास्ट मोमेंट आई कांट से यू टू रीड द प्लांट ब्रीडिंग बुक बाय बीडी सिंह, प्लांट ब्रीडिंग बुक बाय कुंदन सिंह। मैं बोल नहीं सकता हूँ कि हाँ 30 दिन में पढ़ाई कर लो हो जाएगा। द लास्ट ऑप्शन दैट इज लेफ्ट फॉर यू पीपल इज टू गो फॉर द PYQ series, go for the most expected questions and go for the marathon series. These are the 3 success mantra that you can follow right now. पास्ट पीछे को छोड़ दो, लाइक डन इज डन, बट नाउ इफ यू आर लुकिंग फॉरवर्ड व्हाट शुड बी द बेस्ट स्ट्रेटजी। अगर आपने प्रिपरेशन कंटिन्यू रखा है देन दिस विल बी एडिशनल, बट अगर आपने कुछ भी प्रिपेयर नहीं किए हो या स्टार्ट कर रहे हो या कुछ भी मिडल पे हो जहाँ पे आप कॉन्फिडेंट नहीं फील कर रहे हो, देन द गुड ऑप्शन इज, द बेस्ट ऑप्शन इज गोइंग विद ऑल द मोस्ट एक्सपेक्टेड क्वेश्चंस ऑफ एवरी सब्जेक्ट प्लस गोइंग थ्रू द PYQs ऑफ एवरी सब्जेक्ट, एंड लास्टली द अल्टीमेट रेस्क्यू पॉइंट, गोइंग विद द मैराथन क्लासेस। एंड वन गुड पॉइंट अबाउट दिस मैराथन क्लासेस इज द कंप्लीट PDF। जितना भी PDF हम कराएंगे, PDF में ऐसा पता होगा कि सारा हम पढ़ाते हैं कुछ, ठीक है? ये कंपाइल किया हुआ कंटेंट होता है जो मेमोरी बेस के लिए इजी है और कॉन्सेप्ट्स को याद करने के लिए इजी है। एवरीथिंग इन द PDFs will be directly available for you in our courses. See, in our courses, not just the master course, it will be available in the test series course, so that the students who have even taken the test series, even they will be able to just access the content, and from there सारे सब्जेक्ट्स का, सारे सेक्शंस का जब मैराथन आपके पास होगा, मैराथन PDFs होगा, how much like time will be taking to just revise it? Like 4 to 5 days will be more than sufficient to completely revise the entire plant science. माइक्रोबायोलॉजी का भी हमने सोचा है, बट सिंस इट इज माइनर वी आर नॉट कंसिडरिंग, बट स्टिल वील अपडेट यू, बिकॉज इतना सारा स्लॉट्स है नहीं हमारे पास, बिकॉज एवरी सब्जेक्ट हैज टू गो विद द स्मार्ट बोर्ड। स्मार्ट बोर्ड सिर्फ 2 ही है सेटअप हमारे पास। सो लाइक लेट्स सी, बट विल कवर द मेजर कवर करेंगे, एंड 1 or 2 सेशंस विल आल्सो प्लान फॉर माइक्रोबायोलॉजी, विल नॉट लीव इट लाइक दैट। Let's see like what we can do for it. Still looking handsome, thank you. What about accredited lecturer classes? I asked about lecturer classes. Yes, or if you are asking about the master course classes, master course classes are the more than sufficient. Even if you want to secure all India 1, if you complete all the master course classes, then definitely you will be able to secure it. I thought like you were only asking about this YouTube classes. When will be the marathon completed? The marathon we are going to complete before the 3rd week of June. 3rd week से पहले हमने टारगेट बना के रखा है कि मैराथन सारा कंप्लीट कर दे, ताकि लास्ट वीक आपके पास टाइम रहे खुद के लिए, खुद को टाइम देने के लिए, ताकि थोड़ा ब्रेक भी आपको बीच में मिल जाए। ठीक है, ओके। Fine. Okay, guys. So let him start. बहुत crisp and short होगा. But still, it will be very much engaging. Just enjoy this class. And I'll be seeing like how many of you. So how many comments that you will be getting after this class, and how many views you will be getting. How many students are actually loving him to watch on the screen. Okay, good evening. Good evening, one and all. Hello. Yeah, good evening. Okay, we have now discussed 2020, 21, 22 paper. Okay, 3 papers. Now, today we will discuss about 2023 shift 1 paper. Okay, and the remaining is 2023 shift 2 and 24, 25. We will just complete in upcoming days. Yeah, good evening, good evening. नहीं कुछ नहीं कुछ नहीं, वो थोड़ा नहीं, हम वही बताया था ना वो व्यूज के बारे में. फर्स्ट वीडियो तो 2K उसके ऊपर चला गया था, अभी तो व्यूज भी नहीं आ रहा है. ठीक है, मतलब मैं खुद समझ नहीं आ रहा है क्या हो रहा है. ठीक है, इसलिए बस वो ऐसे आके बताया था सर. लेट्स बिगिन. या विल स्टार्ट. Sorry for Hindi is very good now. ठीक है, thank you so much and don't worry. Okay, I will not leave you guys in the middle of the preparation. We will complete each and everything. ठीक है, बट मैराथन के लिए सपोर्ट चाहिए. ठीक है, ये सारे PYQ वीडियो का व्यूज और आपका इंटरेक्शन, उसके हिसाब से हम मैराथन में चलेंगे. आप इंटरेक्शन नहीं कर रहे हैं सही से, व्यूज भी नहीं आ रहे अच्छा से, मतलब आप एंगेजिंग नहीं हो रहा है, मतलब मैं नहीं ले लेगा मैराथन. ठीक है, मैराथन चाहिए तो आप सारे कमेंट्स डाल दो अच्छे से. आपको मतलब क्या चाहिए वो पूछो कमेंट बॉक्स में, हम सब कुछ दे देंगे. ठीक है, अच्छे से शेयर भी कर दो वीडियो सब के लिए. ठीक है, मैं बताया था ना कल, शेयरिंग इज केयरिंग. ठीक है, we will start today's discussion. This is 2023 shift 1 paper. There will be so many questions which are repeating. Okay, so that you will get to know, okay, where they are targeting, the area of targeting, from where they are picking the questions. As I told yesterday also, by analyzing previous year question papers, okay, you will get to know that from where they are taking the questions, so that you can concentrate on those topics, okay, on your subject. So it will be very easy if you analyze the paper. First question, okay, today's first question: which of the following geographical indication is not matched correctly? Basmati rice, North India; फजली मैंगो, महाराष्ट्र; मैसूर बीटल लीफ, कर्नाटका; मलबार पेपर, केरला. बताओ, which is the following geographical indication not matched correctly? B. बताओ, मतलब फजली मैंगो, where it belongs, to where it belongs. Fazli mango got GI tag from which state? Bihar. I am asking about GI tag. From which state? No, not Bihar. Yes, Chinchu, West Bengal. Okay, you know anyway basmati rice. Okay, it's the 7 states. Okay, Himachal Pradesh, Uttarakhand, Jammu and Kashmir, parts of Jammu and Kashmir. Okay, Delhi, Haryana, Punjab, and western parts of UP. Okay, 1, 2, 3, 4, 5, 6, 7. 7 states. Okay, these are foothills of Himalayas. These states will come under the foothills of Himalayas. Okay, for these states, okay, they got GI tag for basmati rice. As I told in the class also, okay, if you grow basmati rice in southern India, okay, you will not get that trade name. Okay, the basmati. Okay, you should grow in these particular states. And Mysore betel leaf, which is very famous. Okay, in case of Karnataka, they got GI tag. Malabar pepper, okay, which is from Kerala. Yes, 7 state. Okay, we will see about Fazli mango. You can see, okay, who will apply for
GI tag, okay, APEDA, okay, your APEDA will generally, sometimes the groups of farmers, okay, the non-profit organization, some local authorities also will claim for IPR, okay, but in this case, okay, APEDA claimed this particular GI tag, that is Fazli mango, okay, certified fruit primarily grown in Malda and Murshidabad district of West Bengal, okay, and in Rajshahi region of Bangladesh also from Bangladesh government, okay, they have also got Fazli mango GI tag from Bangladesh government. But in India, Malda and Murshidabad region of West Bengal got this particular GI tag, okay. Maybe if they are asking about GI tag, they will target the recent ones, okay. It got in 2008, not recently, okay. In Bangladesh, it is they got under 2011, okay. But if they are asking regarding GI tag, they will mostly concentrate on recent ones, okay. You should be aware of recent ones. Alfonso in Maharashtra. Yes. Okay, the next question. An effective germplasm collection is the prerequisite for any successful breeding program. Statement 2. Genetic variability is the root of any breeding program. Batao. Tell me the answer. What about these two statements? Yes, both are correct. An effective germplasm collection is prerequisite for any successful breeding program. Okay, that's why we are having we are having the National Gene Bank, okay, which is under National Bureau of Plant Genetic Resources. We are storing so many germplasm so that whatever the things, if you want to breed for abiotic stress, if you want to breed for biotic stress, we need some sources, right? Some wild germplasms or wild relatives. So we can collect it from the effective germplasm collection. Second statement, genetic variability is the root of any breeding program. Without variability, breeding is zero. Okay, nothing. You can't do anything without variability. Okay, if you go to the field, you are having so many lines. Every lines are giving 10 kg yield. Then what you will do? What you will select? Okay, there should be variability, right? Some are giving some less yield, some are giving high yield. Okay, some are having resistant plants, some are having susceptible plants, so that you can select whatever you need. Okay, that's why variability is the root of any breeding program. Okay, that's why we will create the variability. Okay, we will create the variation by through hybridization, mutations. Okay, these are the sources of variability. Okay, both the statements are true. An example of primary introduction of a crop varieties in plant breeding in India. What about primary introduction? Yes, Roar. No variability, no selection. बताओ इसको बताओ answer. An example of primary introduction of crop varieties. Exactly, your Sonara 64. The question is simple, but you should know what are the other things. Okay, your Sonalika and Kalyan Sona, okay, are the derivative of Sonara 64. By using Sonara 64, they have identified, or they have improved, or they have used this Sonara 64 as a parent in different hybridization program. Then they have derived Sonalika and Kalyan Sona. Okay, these are secondary introduction. Your PBW 343 also derived from Sonara 64. Okay, which is popularly grown in Punjab area. Okay, this is about your primary introduction. What is exactly Sonara 64? It is spring wheat. Okay, as I told, in Indian case, we will grow spring wheat in winter time. Okay, first, what happened? The Semidor of genes, how they came? They first identified in Japan. Okay, the American researchers. Okay, his name is Vogel. Okay, he first identified some Narin 10 lines. Okay, then he introduced those lines into US. Then Narwan Borla got those lines. Okay, they have developed. Okay, they have introduced RHT genes. Okay, RHT genes. RHT genes are present in Narin 10. Okay, they are responsible for dwarfness of wheat. Okay, first they have transferred to winter wheat. Since they are growing winter wheat, But when they are introducing those dwarfing genes into an Indian condition, we need spring wheat. That's why they have transferred those to Sonara 64. Okay, that is spring wheat. Then we introduced those spring wheat into India. Then we have transferred to our varieties. Okay, then we got Sonalika and Kalyan Sona. Larmaraja 64, Maya 64. These are primary introduction. Okay, they are also spring wheat. Okay, what about this match the following? They are, it is some of the out of the boundary question. Okay, see, in 2023 they have asked about some Louis Brown, Lula Rana. Okay, these are out of the boundary questions, but they will ask sometimes. If it is match the following, then you can easily match. But if they ask in any statement and assertion, then it will be very difficult. See. Okay, A is first. Yes, Lula Rana is first genome-edited human baby. Dolly is first mammal cloned. C3. D1 flavor saver is first transgenic tomato. As I already discussed, okay, in the yesterday's class, we are already what exactly the flavor saver tomato. It will produce normal tomato produce polyglucosidase enzyme. Okay, it will do the softening of the tomato. But if we have antisense RNA to that particular enzyme, so it will stop producing that enzyme. Okay, in mRNA stage itself, we can control the production of enzyme so that there is a less production of polyglucosidase enzyme. So fruit will not soften early, so that you can extend the shelf life of the tomato. Okay, they have asked this regarding flavor saver tomato. They are asking from 3 years. I have seen in next shift also in 2023 shift 2. They have again asked this flavor saver tomato. Okay, see how they are repeating the questions. And Dolly. Okay, we will see what exactly Dolly. This is actually the first mammal. Okay, cloned. Okay, what they have did, they took the somatic cell, okay, from the udder of an adult fin dorset. Okay, udder means there will be a gland, okay, the milk glands. From that udder, they have took somatic cell. Then they have transferred the nucleus from that particular somatic cell into the another enucleated, okay, egg cell. This is somatic cell. Okay, this is nucleus. They took this particular nucleus. Okay, they took this particular nucleus from one of the adult fin. Okay, then they have introduced, this is enucleated egg cell from the another ewe. Okay, that is Scottish blackface. This is the egg cell. They took this egg cell and they have introduced this nucleus into this egg cell. Okay, this is enucleated, means they have removed nucleus from the egg cell of another ewe and they have introduced this nucleus into this particular egg cell. Then that cell, the nucleus cell, they have fused with the developed embryos in the particular another ewe, so that they have cloned one of the somatic cell from another sheep into the developing embryo of the another sheep. Okay, this is the exact what they have did in the cloned mammal, the first cloned mammal. Then they will get a sheep, then they have named it as a Dolly. Okay, the Dolly was named after country singer, okay, Dolly Parton, in a public contest. Okay, this is about your Dolly, the first mammal cloned. The next question. It has been observed in some cases that particular quality trait is not expressed as such in the wild species, but it is detected only in the segregants recovered from its crosses with the cultivated relatives. Such traits are known as. बताओ, फारूल आ गया फिर से बीच में. नाइस प्रेजेंटेशन सर. ठीक है पारुल, थैंक यू सो मच. बताओ, अभी बताओ, इसका आंसर बताओ, कमेंट में बताओ। डी 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 latent trait. Okay, for example, inequality traits in case of breeding programs. Okay, in the wild germplasms, for example, in wheat. Okay, there will be so many wild germplasms in case of wheat. Okay, Agilops peltoids. Okay, Triticum caudatum. Okay, then there will be so many different germplasms in case of wheat. So they will not express your quality traits. For example, in case of wheat, gluten strength, okay, high gluten content, okay, we are having in the wild germplasms, but they are not expressing in particular wild germplasms. But when we crossed that wild germplasms, okay, into cultivated one, then we will get segregation generation. That means your F2, F3, okay, in that particular case, they will express. So those traits we will call them as a latent traits. Okay. Next question. Which of the following information cannot be obtained from the progeny test? Very simple. Progeny test. Anyway, you know already what exactly the progeny test. Okay, we will grow the progenies of particular parent and we will evaluate the progenies. Okay, we will see what the traits are inheriting or not. Okay, how they are passing generation to generation. Okay, this is the progeny test. But out of these four, it will not give the information on which of the following. D. Recombination frequency and map distance between the target loci in the plant. It will give breeding behavior of the plant. Breeding behavior is a thing, but whether it is self, whether it is cross, okay, the target trait is heritable or not, it is due to additive or it is due to dominance. Okay, it will also give the heritable nature of the particular trait, the real value of the plant. Okay, if you want to know the genetic worth of parent, okay, we should do the progeny test. Okay, we don't know, maybe there will be an environment influence sometimes, that's why it is giving high yield. So we want to know the exactly the worth of an individual due to environment or genetic factors. So through progeny testing, okay, we can see what exactly the parent is. Okay, but recombination frequency and map distance, ये तो okay, it is not possible through the progeny test. For this, we should go for test cross, okay, or back cross or any mapping population for this particular objective. Okay, अभी बताओ कौन दिया था progeny test? Which scientist gave progeny test and in which crop? Yeah, Wilmorin, Johnson, Wilmorin. अंकुर बता रहे जॉनसन, डिलीट कर दिया मैसेज. Exactly, it is given by Wilmorin. Okay, we will also called as Wilmorin isolation principle in sugar beet. Okay, this is the one part. But as Ankur told, okay, Johansson also give progeny test. Okay, Ankur, you are right. Okay, you are also right. Wilmorin is given the basic principle and he tested in sugar beet. Okay, but the real practical application was given by Johansson. Okay, your pure line theory. Through the progeny test only, he was able to give the principles. Okay, your trait is due to environmental or your the selection within the pure lines is not effective. Okay, these are given by based on progeny test only. Okay, yeah, Johnson gave pure line theory. It is also one of the progeny test, but the basic idea is given by Wilmorin, but the practical applicability and principle is given by Johnson. Clear? Clear? Is it clear? If they given Wilmorin and Johnson, you should choose Wilmorin. But if they are not giving Wilmorin, if the in option contains Johnson, then you can easily go for Johnson. The breeding method usually used to produce the nucleus seeds of a variety of cereal crop, okay, which is actually from the seed tech portion. Okay, but yeah, you can answer. The breeding method usually used to produce the nucleus seeds. Hey. Okay, here to row method. First, the seed, handful of seeds. Okay, the handful of seeds, what we will call to that particular seeds? Nucleus seeds, right? Your handful of seeds are the original handful of seeds which are developed from breeder. Okay, we will call them as a nucleus seeds. How the breeder will produce nucleus seeds in their field, in their own field. Okay, if it is wheat, okay, from single plant, there will be so many tillers. Okay, in particular tiller there will be a year. Okay, to get a pure seed, okay, so that those are the original seeds, nucleus seeds. So from that seed we will produce breeder seed. Okay, so we need to maintain purity. From you know this particular individual, though that is developed by him only. So he will take particular year and he will grow that year in a single row. Okay, single row like that. Okay, from year to row method, they will try to produce a nucleus seed. Then they will give that nucleus seed into the production of breeder seed. Okay, we will only produce breeder seed again, but from this particular row, then they will collect the seeds and they will go for breeder seed production. Okay, from head, yeah, it is based on the different crops. अरे ले लो, फिर से आ गया क्वेश्चन. ठीक है, if they ask this kind of question, okay, you should be able to answer within 5 to 10 seconds. Okay, you just find anyone which is correctly matching, then you can easily complete the, then you can save the time. You can see in C 1, 2, 3, 4. So if you match this C, then you can easily choose the answer. What is C? Heterozygous and homogeneous. Which one is heterozygous and homogeneous here? Huh? Your sorry, heterozygous. Yeah, C, heterozygous and homogeneous. This is your hybrid. Okay, C 1. In all these cases, C is 1 in this particular case. Okay, just save your time and move on. Okay, this is the thing. By this you can save the time. Okay, all other, you know, homozygous, homogeneous are your pure line variety. Homozygous, heterogeneous is your multi-line variety. Heterozygous, heterogeneous your synthetic variety. Okay, this is the basic one. You should easily answer this particular question. Okay, next one is obtaining intellectual property rights in the form of patent is an important activity to protect any invention or discovery. However, filling an application for patent is very critical step in this process. Here the list of the steps, so you can just arrange, okay, from the starting step. Okay, first what you have to do, first for any filing an application, you should first develop a patent product. Okay, after that you can file an application to patent office. B, A. After that, they will scrutinize and publication of your application by the patent office. Okay. After that, they will give for public challenge. If anyone wants to challenge this particular patent, okay, then they can challenge. C, BADC, BADC. Okay. Yes, C is your answer. But we will see what exactly happens, how the patent office will grant the patent in Indian condition, Indian scenario. You can see, first you should develop an invention. It should be novel, okay, and industrial application. Okay, after you develop an invention, you can just specify what exactly it is and you can file an application. Form 1. Okay, you should no, okay, Form 1. After filing an application, okay, they will public, okay, they will made it for public. they will public, they will in their official journal, they will go for publication. The application examined and for formalities and published in the patent journal after 18 months from the filing date for the public knowledge. Okay, but before that also you can approach them for the early publication. They will publish in their original publication. After that, any person file a pre-grant opposition within 12 months from the date of publication. Okay, now I have applied for patent. Okay, any one of my friend, okay, he also doing that one, or maybe he already applied for patent for that particular product. Okay, he can challenge, okay, it is not authentic, or any other reason, for any other genuine reasons. Okay, before 12 months after publication, you should oppose. Okay, then what they will do? They will examine. Applicant request for examination. After examination, they will give an examination report. So, if anything, if we want to give an external additional information for patent office, we should give in this particular specific period. Then they will grant the patent. Okay, patent office accept the application and grant the patent. They will also give certificate. It is valid for 20 years. Okay, for after third year, every year you should go for renewal. Okay, you should remember that your patent will be valid for 20 years. Okay, novelty, inventive step and industrial application, it should be there, and 20 years from the filing date. In Indian condition, the Patent Act 1970 and Rules 2003 are presently processed in India. Okay, Patent Act 1970 and Rules 2003. First, you should develop an invention. Then you should prepare specification what exactly it is. Then you can go for filing an application. Then they will publish in their journal. Okay, if anyone wants to oppose within 12 months of that publication, you can go for opposition. Then there will be a decision whether we should grant or we should suspend. It will be in the hand of patent officer. Then they will grant the patent. 20 years will be your period for your patent. Okay, after 3 years, every year you should pay the renewal fees. This is about patent. Next question. The demerits of mass selection in self-pollinated crop plants include. Demerit of mass selection. They are asking about mass selection. What are the demerits? Batao. You can see the options. You just have to leave one of the options. Yeah, D. Yes, PCD only. It is not possible to know phenotypic superiority of selected plants due to genotype or environment. We are only seeing what. First, I will tell what exactly mass selection. You will go to field. Okay, there will be a maize field. For example, you just see phenotypically of all the plants. You will just select the best possible superior phenotypes, and you will take those plants and you will mix the seeds from all the plants, and you will grow the next generation. Okay, this is simple mass selection. From this, you will not get to know that your superior phenotype, it is due to genotype or phenotype. You are not going for progeny test also. Okay, you are just bulking the seeds. Okay, this is the one of the disadvantage or demerit of your mass selection. Varieties selected through mass selection are not as uniform as pure line varieties. As I told, you are mixing the seeds after the selection. So there will be a impure zygosity. It is homozygous or heterozygous of the selected plants is also not known. Okay, if it is heterozygous dominant and if it is homozygous dominant, they will both are giving superior performance. We are not able to differentiate both of them. If you are doing progeny test, okay, your heterozygous dominant will segregate in the next generation. AA, small a, small a. So you can see the segregation in the progeny test, so that you can avoid this particular plant. Okay, but in mass selection we are not doing that. Okay, so that these three are demerits. Varieties developed by mass selection are quite easy to identify in the seed production program. It is merit actually, as I have told. Okay, it is based on completely phenotype. So, for example, in maize, you are selecting for the cob length. The cob length which is giving 15 centimeter or more, you are selecting for the developing in a variety. In the seed production plot, you can easily identify. For example, we can take another any simple phenotype. The anthocyanin pigment in the stem. Okay, you are selecting based on the anthocyanin pigment in the stem in maize plants. Okay, in seed production plot, okay, whatever which are absent of that particular anthocyanin pigment, you can easily remove. Okay, that's what the merit in case of mass selection. It is completely based on phenotypic selection, so that it is easy to identify in the seed production program. Okay, easy because only selected the phenotype only. Yes, Vijayali. Okay, selection based on phenotype. That is only demerit. So merit. All other are demerit. So this is about our course. Once again, I am telling. Okay, whenever not purchased it, you can purchase at 1,999. Okay, you can cover all the syllabus. Okay, in 30 or 40 days. Maybe it will be helpful for your exam. For next year students, okay, one who are watching this in our YouTube channel, okay, for next year we are having mock test bundle course which is available at 799, and our course, okay, for next year exam 4,999. For 2028, okay, we are having at 8,999. Okay, this will be very beneficial for you guys, as I am telling in each and every video. If you want any feedback or suggestion, you can ask your seniors who are already enrolled in our courses. Okay, they are having some better idea, so they will guide you guys. Okay, that's what my suggestion is. Fixation of atrocities, ले लो, फिर से ले लो 4 marks आपका जेब में. ठीक है, very simple one. We have already discussed it. Okay, बताओ, आंसर बताओ. Fixation of atrocities cannot be achieved by. Yes, exactly, pure line production. Okay, and if I ask the question, fixation of atrocities can be done through polyploid breeding. The statement 1. Statement 2, fixation of atrocities can be done by allopolyploid. Statement 3, fixation of atrocities can be done by autopolyploid. Which statement is true? Fixation can be done by polyploid, autopolyploid, allopolyploid. Allopolyploid. Good. Okay, we are, as I already told in the yesterday's class, your allopolyploid are from different genomes, so that they will show the atrocities in the one or the other way. Yes, good. Allopolyploid. When several genes are considered together, the proportion of homozygous for all the genes in the backcross generation in self-planted crops given by the formula. Batao. For homozygosity, when there are number of genes and the number of backcrosses. Intragenic, intraspecific cases, they will compensate genomes. D. Exactly. Okay, 2 to the power of m minus 1 divided by 2 to the power of m whole divided by n. M is your, what about m? Is your number of backcrosses. Okay, and also for selfing, the number of selfing you are going. Okay, for both number of backcrosses and selfing, you can consider m. You can just replace m with the number of backcrosses or selfing, and n is your number of genes. It is completely dependent on your number of genes. Okay, how many genes are segregating together. Okay, this is your formula for homozygosity. For heterozygosity, it is simply 1 divided by 2 power m for heterozygosity at any backcross generation. This is for homozygote. ठीक है. VK AK 4 VK. One like for amazing teacher. Genetic homeostasis, which confers a population wider adaptability, is however not exploited by genetic homeostasis, okay, which will give the population wider adaptability, but which is not exploited by which of the following breeding outcome. बताओ, इसको बताओ, आंसर बताओ. A, B, hybrid, A, B. In question itself, it's there. Population wider adaptability. Okay. Your hybrids will not exploit the genetic homeostasis. Genetic homeostasis is nothing but it is buffering capacity of an individual for the changing environment, so that it will give wider adaptability. Wherever you will try to grow, they will give some good performance. It is due to genetic homeostasis. Okay, but in case of hybrids, they are homogeneous. They are homogeneous in nature. Okay, so that we can't exploit the wider adaptability in that particular case. Okay, we will try to target particular environment in case of hybrids. But varietal mixture and multiline varieties, synthetic varieties, composite varieties, these are heterogeneous in nature, the population, so that we can exploit wider adaptability. ठीक है. Resist to change, yeah, that's what I told. Okay, buffering capacity to changing environment. Okay, they will try to adapt for the new conditions, new environment. Okay, apart from hybrid, all other are heterogeneous in nature at population level, so they can adapt to the changing environment. Okay, अरे ये तो फिर से आ गया. कल भी हम डिस्कस किया था. देखो क्वेश्चन हाउ दे आर रिपीटिंग. Okay, regarding, as I told, these are very important. Your CBD, ITPG, RFA, Cartagena protocol, Nagoya protocol, NBA, PPFRA. Just make it, note it down. Okay, you just go through those important acts. You can just match the following year. Very simple. Okay, there is no need to think too much about it. Your CBD will promote conservation and sustainable use of biodiversity, which is 1993 came into force. Your ITPG RFA 2004. Okay, ensure conservation and sustainable use of plant genetic resources for food and agriculture. We are already discussed about yesterday. Okay, which are the crops they have covered. Okay, how many food crops, how many fiber crops, what are their main objectives. Okay, the one who want to know about ITPG RFA, just go back to the previous class, then you can get some idea. Cartagena protocol. Okay, this is comes under your Cartagena protocol and Nagoya protocol. They will comes under CBD only. Okay, your Cartagena protocol came into force in 2003. Your Nagoya protocol came into force in 2014. Okay, Cartagena protocol will ensure safe handling, transport and use of LMOs, and your Nagoya protocol access to genetic resources and fair and equitable sharing among the different countries. कौन सा option? For A4. Okay, in first option itself we will get an answer. For A4, there is only one option which is having A with 4, so you can easily, yeah, 64 crops, exactly Kavita. In that 35 are food crops, 29 are forage crops. Cartagena 11 September 2003. Yes, 2003. Nagoya protocol 2014. Yes, Ankur, good. Okay, first, but the Cartagena protocol, it is given means in the 2000, but it came into force in 2003. Your Nagoya protocol 2010, but came into force in 2014. फिर से आ गया ये भी. See how they are repeating, okay, so that you should know these are very important. National Food Security Act, that is 2003 for A, 2, PPFRA 2001 for B, 1, okay, Biological Diversity Act, that is 2002, C, 4, this is your answer, okay. The National Seed Act, which is 1966, yeah, as I told yesterday, PPV FRA 2001 came into existence in 2003, okay. 2005 authority, 2006 rules, 2007 the first variety registered, okay. This is about different years of your particular question. Here you can see, match the following. बताओ. Again, it is very simple regarding dwarfing genes. B. Yes. Rice, DG Ujan, wheat, Norrington. These are the sources, okay, the varieties which are having the particular genes. In case of wheat, your RHD gene, it is dominant, okay, the dominant mutation. But in case of rice, SD1 gene, this is recessive gene, recessive mutation, okay. These are the responsible for your dwarfness in case of wheat and rice. The source is in wheat, Norrington, in rice, DG Ujan, for maize, CMST, that is cytoplasmic male sterility source, for pearl millet, TIFT 23A. Okay, as I told, your RHD genes, that is Norrington, identified in Japan, then it moved to US, then CIMIT, Mexico. From that, for our Indian condition and Pakistan. Pakistan also they have introduced as a Mexi-Pack. Okay, Mexi-Pack. In case of rice, it is identified in Taiwan. Then they have developed a TN1 variety, that is Taichung Native 1. Then it came to IARI, Philippines, IR8, IR24, IR36. Okay, these are the mega varieties of rice which are semi-dwarf in nature. What about the answer? For rice, DG Ujan 3. There is only one option, okay. Your answer will be B. SD1, SDN2, yes, maize, Texas cytoplasm, mutated DELA protein, yes, Jana. Okay, your RHD gene is responsible for mutated DELA protein, so that it will not do the gibberellin to activate, so that you will get a reduced diet. In case of SD1, it will, mutated SD1 will not produce the gibberellin oxidase 20, so you will not get an length of your particular crop plants. There will be dwarfness. Okay, plant height will be dwarf. Arrange the steps of developing synthetic variety of a crop. Again, synthetic variety, as already we have discussed yesterday. Okay, first we have to start with identification of suitable parental lines. Okay, after that, growing of, mixing of equal amounts of seeds from the parental lines. In synthetic, particularly in synthetic crops, identification of suitable parent lines based on GCA. Okay, based on GCA. We will test for GCA, but in composites there is no GCA. The same process. After that, you will mix the equal amount of seeds from the parental lines. B, A. Then growing of mixed seeds in open pollinated condition. You will leave for open pollination. Then multiplication by open pollination for one or more generation and distribution to the growers. D, BACD. Clear? The only difference between synthetics and composites, you will test for GCA. In composites, you will not test for GCA. So that again, to produce synthetic seed is very easy. Okay, to make that same combination. Okay, you have some lines and you are tested for GCA. But in case of composites, it is very difficult to reconstitute that particular product. Okay, since we are not tested, we will just mix the seeds. Okay, from the different inbred lines. Okay, So that in synthetics, you can grow for 4 to 5 years, but in composites, you can grow till only 2 to 3 years. Okay, after that, you should again purchase new seeds. The main feature of idiotype proposed by, idiotype proposed by Donald for wheat include. Okay, your idiotype breeding is given by Donald in 1968 for particularly wheat crop. Okay, out of these, which are the main features which are included? You should remove only one option. A, B, C only. Okay, absence of ants. This is incorrect. You should have presence of ants. In wheat, okay, in wheat, if I say the 40% of photosynthate, photosynthetes are nothing but the grain filling. Okay, the whatever you will see wheat grains, the 40% of photosynthetes will be produced through the ants. Okay, ants are nothing but this is your spikelet in your wheat spike. Okay, this will be your ants. If you see, this will be your ants. The most of photosynthetes, okay, which will accumulate in the grains through the ants. So that Donald will include this presence of ants also in case of your idiotype. Okay, single short strong stem, erect leaves, long ear, and presence of ants. Okay, the answer is A, B, C only here. Okay, there is absence of ants is not correct. Presence of ants is correct. On photosynthesis also, yes, that's what I am telling. If there is no ants, then it will reduce the photosynthetes. In rice, flag leaf, yes, flag leaf will more accumulate the photosynthetes. Which of the following molecular marker techniques is used best for studying genome-wide genetic variation in croplands? Simple. मैं क्लास में बताया था already. I have already discussed about this in class, in our markers class. D. Okay. Answer will be D. SNP is why. If I ask why SNP is best for studying genome-wide variation in croplands, what are the reasons? लगा दो, comment में लगा दो अभी. What are the reasons why we are using most of the cases at present scenario we were using SNPs? What are the reasons? Yeah, comment box. Yeah, single nucleotide, codominant, biallelic, high throughput and reliability. Yes, Sinchan. This is the main codominant. We are having SSR markers. Okay. For biallelic, we are having different markers. High throughput is the answer. Okay. At the single run, you can go up to, as I told, chips, SNP chips, 35,000 markers you can analyze at a single run. Okay, 80K chip, 90K chip in case of wheat. Okay, you can see high throughput and well automated. These are the two important reasons. Okay, high throughput and genome coverage. Okay, it will whole genome availability. Okay, genome coverage. These are the two reasons for why we are using SNPs at present days. Which of the following is true regarding CRISPR-Cas9 gene editing in crops? Only two questions are there in this particular session. Yes. Which of the following is true regarding CRISPR-Cas9 editing in crops? It involves the insertion of foreign genes into the crop's genome. It can only be used to delete genes, not modify them. Okay. It allows for precise and targeted modification to a crop's DNA. It is new technology but not for use in crop improvement. अरे ये तो crop improvement के लिए use कर रहा है, ठीक है. Allows for precise and targeted modification. That's what our CRISPR will do. It can only be used to delete genes, not modify them. Okay, this is the not correct. It involves the insertion of foreign genes into the crop genome. Okay, sorry, they are asking about true. It involves the insertion of foreign genes into the crop genome. We will also edit. Okay, we are also delete the function of gene. It can only be used to delete the genes. It is also not correct. It allows for precise and targeted modification of crop's DNA. It is new technology, not for using crop improvement. Okay, what I have told, it is for crop improvement. Okay, the correct is the C option. Okay, the precise and targeted modification. As we already told that SDN1, SDN, SDN3. Okay, there will be one question when I am preparing regarding CRISPR. I will clearly explain again in the tomorrow's class. Okay, regarding SDN1, SDN2, SDN3. But at this point of time, you should know it allows precise and targeted modification of a crop DNA. Okay, it not only insert the additional gene, it not only delete the genes, okay, or modify. We can do each and everything in CRISPR-Cas9. We can edit, okay, we can delete, we can add the new gene. Okay, these are the different things in the CRISPR-Cas9. The consideration that prompted the development of the plant breeders' right system in India. Okay, it includes, what are the consideration for PBR rights? The development of a crop variety is an innovation and it is intellectual property which should be protected. It encourages plant breeders by providing economic incentives. It encourages the private sector to invest in the plant breeding activities. Plant varieties are not absolutely stable, hence need protection. A, B, C only. Your D. Okay. The main criteria to protect your variety under PPFRA is N does novelty, distinctness, uniformity, and stability. You can see this is the basic criteria to protect the variety. They are telling plant varieties are not absolutely stable. Okay, if they are not stable, then it is waste to protect that particular variety, right? Okay, so that D is not a correct option. Your A, B, C. Okay, it will, the development of a crop variety is an innovation. Okay, you can protect as a intellectual property. Okay, it will encourage the plant breeders by giving breeders right, so that they will also get some economic help. Okay, they will also get some economic incentives, royalty, they will get royalty. It encourages the private sector to invest plant breeding activities. Okay, when we are protecting the variety, so that no one will sell publicly, so that particular variety will be sold by that particular breeder, original breeder, so that they will get some profit. So private companies will encourage us to the breeders to development of a variety. Okay, they will, it is indirectly the encouragement to the breeders, to the private sectors. Okay, this is your answer. Okay, this is all about today's session. Okay, you can see there are so many questions they are repeating, but I am taking those questions also in this discussion session. Why? Just I want to tell you guys that, okay, these are very important. If they ask once again in your exam, you should be able to answer, and there is no way to make a mistake. Okay, so that you can avoid you can save your time, you can invest that particular time into another questions, okay, which are from confusion, okay, some analyzed part of your exam. So you should be very careful regarding these kinds of questions. Okay, and you will also, you are now you are getting, by seeing last 4-year paper, you are already got an idea that in plant breeding where they are concentrating, where they are targeting. Okay, if I want to say from my side, okay, you can see they are asking about from first part through the sexual reproduction and asexual reproduction. After that, they are targeting from sterility, self incompatibility, then directly from breeding methods, okay, different breeding methods. Then they are asking about hybrid breeding particularly. Okay, you can see how they are asking, and also they are targeting about your IPR part. Okay, you guys will always ask, sir, what about abiotic stress breeding, biotic stress breeding? Okay, they will not target too much about it. If they are asking also, they will ask principles and methods. Okay, in case of your stress breeding, okay, they will not ask you the recent, they may ask recent varieties, but not old varieties or whichever they are present in your BDC book. Okay, you should know some present idea about regarding those things. So don't waste your time in the unnecessary areas. Okay, this is the time to revise the things very properly, and only the part which is very important, you should invest your time in that particular part. Yes, today's questions were very well. Jeevan H. Thank you, Jeevan, for your comment. Sir, most expected series already it is doing from motion, right? Intellectual property rights, 1 or 2 questions in every year. Exactly, Vijayali. Okay, they will definitely ask you that particular questions. Thank you, sir. Thank you, everyone. Okay, we will see the 2023 shift 2 paper in the upcoming classes. Okay, and we will finish in 2 or 3 classes of this particular session, the PYQ series. Okay, thank you. Then good night. Okay, come soon, sir. कहाँ आया भाई जीवन?